Now we'll start with the nucleic acid. How many types of nucleic acids are there? Two types, DNA and RNA. These are the two types of nucleic acid. Now, what are these nucleic acid made up of? They are made up of sugar. Which sugar? It is your pentose sugar. Ribose. Then it is having nitrogenous base. Nitrogenous base. How many types of nitrogenous base? Two types. Purine, pyrimidine. How many types of purine? Adenine, guanine. Pyrimidine, two type, thymine and cytosine. And it is having phosphoric acid. Now, nitrogenous bases are heterocyclic ring compound. They are heterocyclic ring compound. Why we say it? See carefully. If you see purine, I am drawing the purine right now. Purine is 2 ring and 9 membered, in which these positions are occupied by the nitrogen. Remaining carbon is present, but at this position, nitrogen is present. I think so in the chemistry, remaining will be carbon. If entirely carbon, then not an issue. But if suppose in place of carbon, nitrogen is present, then we call it as heterocyclic ring compound. This is purine. And in case of pyrimidine, it is only 1 ring I will draw with the nitrogen at first and third position. Remaining is occupied by the carbon. Now see from why I'm teaching you all these things. See here, your NCERT. This is purine. Nitrogen at first, third, 7th and 9th position in the ring. So this is purine. How many ring? 2 ring. How many membered? 9 membered. 2 ring and 9 membered. Start from here. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 membered. This is pyrimidine. Pyrimidine, if you see, it is 1, 2, 3, 4, 5, 6. So pyrimidine is 1 ring and 6 membered. So from here they can ask you the question. And heterocyclic, because if you know, I'll focus it also. Nitrogen is present in the ring only. See, this nitrogen is of the ring part. These nitrogens are part of the ring. You check in your NCERT, you will find it. That is why we say nitrogenous bases are heterocyclic ring compound. Theory, if you see here, it is given. Living organisms are having number of carbon compound in which heterocyclic rings are present, and these are nitrogenous bases, which are adenine, guanine are your purine, cytosine, thymine are your pyrimidine in DNA, and uracil is a pyrimidine present in RNA in place of thymine. Is when sugar combined with nitrogenous base, you call this as nucleoside. Nucleoside. How to learn? Sugar and nitrogenous base. Now when sugar plus nitrogenous base plus phosphoric acid, then all 3 are there, then you call it as nucleotide. And DNA is a polymer, means nucleic acid is a polymer of nucleotide. And nucleic acid is a polymer of nucleotide. Now, if you have studied in the molecular basis of inheritance between sugar and nitrogenous base here, which bond is present? You will say N-glycosidic linkage. N-glycosidic linkage. Between sugar and phosphate, which bond is present? Okay, because you know, nitrogenous base will be attached to the sugar. Let me draw the diagram here. So this is your sugar and this is your nitrogenous base. So you are saying this is your N-glycosidic linkage is present here. Now it is attached with the phosphate to make a nucleotide attached with the phosphate. So which bond is present? This bond is known as phosphoester bond. Phosphoester bond. So in DNA, to make one nucleotide, you are using two bonds. One is glycosidic linkage, another is a phosphoester bond. For this, I'll show you just 1 minute. See, beta, a nitrogenous base is attached to the OH of first pentose sugar through N-glycosidic linkage to form nucleoside. So nitrogenous base is attached to the sugar. Which carbon? First carbon of the sugar. The first carbon of sugar is always used. And now, if you see in the diagram, check adenine is 2 ring. This is from your 12th class. Pyrimidine is 1 ring, they are showing it. Carefully check it. So with carbon is always first position is used. Okay, carbon first position is used. And nitrogenous bases, 9th position is used in case of purine, and first position is used in case of pyrimidine. So you can draw this way with purine and sugar. It is first and 9th position. Sugar with pyrimidine, it is first and first position. This line means glycosidic linkage. So this is the way you please learn it. To form nucleoside. If it is in the DNA, you have to add word deoxy. If you are not putting deoxy, it means you are talking about RNA. Because you know 2 types of nucleic acids are there, DNA or RNA. Is it clear? Just like deoxyguanosine for DNA, guanosine only for RNA. Same concept is given in your biomolecule also. See here, beta. Same is given here. When attached to the sugar, they are called nucleoside. Example, adenosine, guanosine, thymidine, uridine, cytidine, they all are your nucleosides. They have given it with respect to the RNA. 12th class NCERT has given us with both DNA as well as RNA. Clear? Now, if suppose phosphate group is esterified, ester bond to the sugar, then it is called nucleotide. Phosphate group means phosphoric acid. So phosphate group is esterified to form nucleotides. So beta phosphoric acid is added, that is why we say adenylic acid, thymidyllic acid, guanylylic acid. All these things you please learn it. Okay, beta. And what is nucleic acid? Nucleic acid are nothing. They are the polymer of nucleotides. Example of nucleic acid is DNA and RNA. Is it clear? And DNA is working as a genetic material, though RNA also work as a genetic material, but in few viruses. So I think so this paragraph of NCERT is clear to you. Now, if you see in the diagram, see here at the first position, nitrogenous base is attached. Here also first position, nitrogenous base is attached. And at the 5th position, phosphoric acid is esterified. So this bond, you remember, it's an N-glycosidic linkage, and this is your phosphoester. Clear, beta? Now, see, if you will find all the points we have covered. Now, see here nucleic acid. So, along with the polysaccharide and polypeptide, they are also macromolecules. And for nucleic acid, the building block is nucleotides. And nucleotides is having 3 components. One is your heterocyclic compound, and heterocyclic compound in nucleic acid are nitrogenous base. All these are there. What are adenine and guanine? They are substituted purine, whereas remaining all are pyrimidine. The sugar which is present in the polynucleotide is ribose, means it's a pentose sugar. Or at the 2nd position, oxygen is not there, then we say deoxyribose. So RNA will make use of the ribose sugar and DNA will make use of the deoxyribose. So one heterocyclic compound and second is monosaccharide is clear. 3rd component is phosphoric acid, and phosphoric acid is esterified. So nucleic acid, which is having deoxyribose sugar, is called DNA, which is having ribose sugar is called RNA. So I think so this paragraph, if you check, this is also clear. Now see here. To make a nucleic acid, a phosphate group is attached to the 3rd carbon of one sugar of one nucleotide to the 5th carbon of the sugar of the succeeding nucleotide. And this bond is also ester bond Two ester bonds are there. That is why we say diester bond. Now let me explain you what do you mean by this thing. See here, try to understand this diagram. This is your first carbon, second, third, and this is fourth, and this is fifth. Clear? Same here, first, second, third, fourth, and fifth. So this fifth was holding the phosphate group. This was ester bond. Now this is your one nucleotide. This one nucleotide is making the diester bond, again ester, second time ester is diester, with the third carbon of the succeeding nucleotide or not. See this line carefully and then read. Phosphate group links the third carbon of one sugar of one nucleotide to the fifth carbon of the sugar of the succeeding nucleotide. And this bond, which is present between the phosphate and the hydroxyl group, is again ester bond. Two ester bonds are present. That is why we say diester bonds are present. Clear? Let me explain. This I am drawing it. Adenine. I should remove the H group because I am discussing the DNA. The second position of oxygen is not there, only hydrogen. Here OH is there. This is your ribose sugar, this is nucleoside, and now I'm adding the phosphate group. Remove the water molecule. So this is your ester bond is made. This is one nucleotide. Let me draw the second nucleotide. And I hope now I can be fast. You can understand it also. H, OH, H, CH2, O, P. Can I go this way now? I hope you are able to follow me. Already the ester bond, I have shown it by removing the water molecule. This water molecule is removed and what is left O, I have shown it in my diagram. Let me draw the third one also. This much is clear on my board right now. I have drawn 3 nucleotides. Now see, I have to join them. 2 nucleotides I have to join. And how will I join? Again by removal of water molecule, means condensation reaction. But between whom? Between the third OH and this one. This is diester bond. This is diester bond. Properly explained in the 12th NCERT, see beta. Properly explained. Two nucleotides are linked through 3:5' phosphodiester linkage to form dinucleotide. Is it clear? 3:5' phosphodiester linkage. Please remember this is phosphodiester linkage. 3:5'. And see here carefully. When a phosphate group was attached to 5th carbon of a nucleoside, then we say it is ester linkage and you are making nucleotide. To make nucleotide, ester linkage. To join the nucleotide, second time ester, we say diester linkage. Understood? Now from this one question I can ask you. If I ask you, first of all, tell me how many nucleotides are there on the board? You will give me answer 3 nucleotides are there. Means I can say nucleotide means bases. In the short, we can say base also. So if number of nucleotide right now I've drawn 3, so you will say 3 nucleotide means 3 bases. And how many glycosidic linkage? You will say how many glycosidic linkage? You will say 3 glycosidic. 1, 2, 3. The glycosidic linkage is equal. How many ester bond? You will say 1, 2, and 3 ester bond. But how many diester? Diester is only 2. So for formula for diester is base minus 1. This is your answer. And if they ask you question, I am having 1000 nucleotides, single-stranded DNA. How many diester can be there? Then minus 1, it means 999. Now, if I have to draw the second strand, see the second strand. So this is your OH, H, and here T. CH2O, P. I hope now you are able to understand, so I can be fast also. Now I have to make diester. This is diester. And in between them, I have to draw hydrogen bonds. How many base pair I have drawn? I will say I have drawn how many base pair I have drawn? 3 base pair I have drawn. It means total bases are how many? Total bases are only 6. 6 bases. How many diester is there? Obviously, you can see diester is 4 only. The formula for this, now see formula for base pair. If they give you base pair, then base pair multiplied by 2, whatever total you are getting, minus 2 will be your answer. So here if you see total was 6 minus 2, 4 is your diester. And diagram is also showing you 4 only. Now if you see this, beta see carefully, this OH is not used. And here this OH is not used. Same, this OH is not used, this OH is not used. If you see this OH is present at which carbon? It was of the 5th carbon. So we call this is as 5th end. If you see this OH is present on which carbon? You are saying it is on the 3rd carbon. So you are saying it is 3rd end. Check this. Clear, beta? Now if you see this OH is present of which carbon? It is present on the 3rd carbon. And here 5th carbon. That is why we say the 2 strands of DNA are anti-parallel. If one is running from 5' to 3', another is running from 3' to 5'. And I hope so now your entire picture might be clear to you. All the numerical questions which they ask, I have covered it also. Now see one more thing. See, DNA. Uniform diameter of 2 nanometer is maintained. Uniform diameter of 2 nanometer is maintained. And if you see, this is your major group. This is also major group. The distance between one major to one major. We can take one minor to one minor also. This is one minor, this is one minor. I'm taking major to major. Is known as 1 turn. And 1 turn will have roughly 10 base pair. It's of 3.4 nanometer. It means distance between one base pair is 0.34 nanometer. Now see carefully. First, let us cover the 11th class. See the biomolecule. Nucleic acids are showing secondary structure, helix. Okay, and the helix is actually double helix. Who has given us? Watson and Crick has given this. So we call it as Watson-Crick model. It's a double helix. The 2 strands are anti-parallel. I think so. I try to explain you run in opposite direction. If you see one is running from 5' to 3', another is running from 3' to 5'. Is it clear? In one case, O is up for the sugar. In second case, O is down for the sugar. The backbone, who is making the backbone? Backbone is made by sugar phosphate, sugar chain. And nitrogenous bases are projected perpendicular to the base, but inside. So beta, if I say it is inside, to try to understand these bases are hydrophobic or not. And phosphoric acid is outside because phosphoric acid is hydrophilic. Is it clear? And you know very well that A pairs with T and G pairs with C. So A and G of one strand always pairs with T and C of another. A pairs with T and G pairs with C. Between A and T, 2 hydrogen bond. Between G and C, 3 hydrogen bonds are there. So 2 hydrogen bond between A and T, 3 hydrogen bond between G and C. It is a helical staircase. Each step represents a base pair. At each step, it turns 36 degrees, and it means for one turn, for one full turn, it means 360 degrees, 10 base pair will be covered. Will involve 10 base pair. And if you see, one base pair distance is 3.4 nanometer or 34 angstrom. So it means per base pair is 0.34 nanometer or 3.4 angstrom. Either you say 3.4 nanometer or you say 34 angstrom is same thing. Which we have discussed is BTN. Now see in the 12th class beta molecular basis. We have already studied this thing. It is made up of 2 polynucleotide chains. Backbone is sugar phosphate and bases are projecting inside. They have anti-parallel polarity. One is running from 5' to 3', another is from 3' to 5'. The 2 chains are in right-handed fashion. Please focus, beta, because if you remember, we studied protein also yesterday. Helix, double helix, that is also right-handed helix. Pitch is 3.4 nanometer, which is having roughly 10 base pair. Roughly, it is not exactly. Here they can confuse you question. It is roughly, not exactly. Is it clear? And 1 nanometer means this much meter. So you have to learn these values. As a result, distance between one base pair will be 0.34 nanometer. See here, beta, in the sugar, this is OH is up. O is up right now. This center O is up. The center O is up here, and here it is this way. Is it clear? Here third OH is left, here 5th phosphate, and here again third OH, here 5th phosphate. This is what? This is your diester bond formation is going on. This is diester bond formation is going on. Is it clear, beta? So I tried to cover this topic. So just see your NCERT. I think so. Biomolecule, up till here, each and every concept is clear. Only the